Okay, everyone, thanks for coming to this week's Physics Colloquium, what used to be called the Physics Research Conference until it got demoted this year. We're very happy to have Sara Walker from Arizona State. I was going to read off all of her titles. There's a long list, but they're right there. Sara, like many people, started life as a cosmologist. She got her PhD in Dartmouth, and she arrived looking to work with Marcello Gleiser on early universe cosmology, phase transitions, and things like that. But then Marcello became interested in astrobiology, and that changed your life. Is that fair to say?
It's true.
That's right.
Dramatically changed it, but not really.
The first famous physicist to start out as a physicist and then make an impact in the study of life was Schrödinger, right? Who wrote this wonderful little book called What is Life? And Sara's going to be telling us about life is what, so maybe that answers the question, I'm not sure, but we were just talking about this earlier today, but for the young people in the audience, this is just a wonderful area where there's clearly a lot of physics, a lot of mathematics and computation and information theory involved, as well as a lot of biology, planetary science, geology, things like that. And unlike particle physics or cosmology, there's no standard model. We don't know what the right answers are or even where to start. So there's a lot of low-hanging fruit to be picked. The difficulty is not, the difficulty is figuring out what questions to ask and where to start answering them, and therefore asking life is what is exactly the right place to start. So we're very happy to have Sara here to answer this question for us today.
I'm actually... Thank you. I'm really happy to be here. It's been a lot of fun talking to people today. And I can't make any promises I'm actually going to answer the question. I just want to ask it. And part of my reason for phrasing it the way I did is that I think when we do actually figure out what life is, it's going to be deeply counterintuitive or not obvious. And being trained in physics, I think about it sort of the same way that we developed theories in the history of physics. We found, reality is not at all the way that we might have thought it was structured. For example, who thousands of years ago would have thought that gravity, the fact that I'm stuck to the Earth, is because space-time is curved, right? There's like this really bizarre structure to reality. And life, we don't actually understand what physics underlies that. So I think there will be a point of declaration or surprise when we actually figure it out. So because the question, what is life, is so hard, or life is what, whichever way you want to phrase it, I'm going to start with some philosophy in my talk, and I think it's because it's deeply important to get the questions right, and I want to give some background on how I think about the problem, having come from theoretical physics and being trained in cosmology, and thinking about it a little bit differently than people in my field typically do. So I work in origins of life and astrobiology. People in those fields are interested in how life emerges from chemistry and what kind of biosignatures we might look for on other planets. And what I'm interested in is what is life as a fundamental process and how do we understand what life is. And to me, that question is not just a question about chemistry, it's a question about how we get things like us and what is the distribution of matter like us in the universe. So what is intelligent matter, what is living matter? So I think of it very much like a cosmologist would. And in some sense, the argument is that there should be universal laws that underlie biological systems, and somehow they should allow us to predict how much of the universe actually becomes alive, or what are the limits of living processes. And so that's sort of obviously a big question. And it's been asked a variety of ways by different people. I think in theoretical physics, we do have some assumption that most of reality is comprehensible, and we can write down theories to explain it. And so I like this quote from Albert Einstein, where he makes the claim that theoretical physics should be able to explain everything, including life. But obviously, we don't have an explanation for life yet. Some people might argue that life doesn't need a deep explanation, and I'd be happy to have that discussion, but I'm very much obviously already stating my biases that I'm on the other side of it, and I think there are laws or some kind of theoretical physics that will explain life. So the first question I want to talk about is Schrödinger's question, what is life? And so Sean already alluded to this book that Schrödinger wrote, which was based on a series of lectures he gave in 1943. And he was deeply interested in what physics and chemistry could contribute to the question of understanding what life is. And so one of the questions that he asked in that book, or the main question of the book, is how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? All right, so this is a basic question you can ask. How many people here have read that book? I'm just curious. Okay, a few people have. It's a really great book. It's an interesting set of intellectual exercises. A lot of it is more about the logic of consistency of physics with what we see in biology, and then you can make some deductions about what kind of processes or physics could actually explain biology. So, for example, he was able to predict the structure of DNA as an aperiodic crystal because it was the only kind of structure that could encode enough information to specify a living organism. And then that was obviously discovered later as DNA. But what he does toward the end of the book is he kind of takes a turn, and this is where I get into the life is what bit of it, that living matter, while not eluding the laws of physics as established today, is likely to involve other laws of physics. So he makes a speculation that maybe we just don't know sufficient physics yet, or there's some other physics that we don't have in sort of our corpus of theories that might actually explain life. And so he does make this speculation, and then the question we're left with is what will ultimately explain life. And as I stated in the introduction, I think it's going to probably be deeply counterintuitive, but I think there are some suggestions about what that kind of physics needs to be. And this is where we start getting into a little bit of philosophy about how we should think about the problem. Because obviously as physicists, we're interested in descriptions of the world that are, have a lot of explanatory power, are unifying in their approaches, and people have told me various things about this hierarchy of theories. I don't so much care about the hierarchy of theories or what their ordering is or how we think about them as the fact that if we want to think about a theory of everything, we have to consider the fact that our current theories of everything are theories of everything except those things that theorize. And this is a quote from David Krakauer at Santa Fe Institute, but I think it does kind of get to the point that we've gotten really far in our understanding of the physical world, but we haven't really gotten to the point of explaining us. And so in physics, we think about that as the problem of the observer. In thinking about physics of life, it's really about how do we actually explain systems that can extract information from their environment and understand their environment, and even do things that we do scientifically, like construct theories of the world. So if I wanted to start to try to think about what that physics might be, the way that I think about it is really that life is somehow related to information, and there is like a deep history, obviously, of people thinking about information somehow being fundamental. And so Wheeler has this famous dictum, it from bit, that all things physical are information theoretic in origin, and we do have information theory as a theory, right? There is actually a rigorous mathematical theory of information that Claude Shannon developed, but I don't think this is quite sufficient to explain life, and that we haven't really gotten to the idea of what information is as a physical process, and that we really need to understand that. So to get to the question of what is life and how do we think about it, I think a couple thought experiments or ways of kind of probing that physics are really useful. And so one I usually kind of use as one of my default ways of explaining how I think about it, which may or may not be the right way of doing it, but I think it's incredibly illuminating about how we can think about how to start probing where we actually need to develop this kind of new physics, if it exists. Is to think about something as simple as the fact that our planet is launching satellites into space. So we don't usually think about the physics underlying that process, except for the fact that we know that we have theories of gravitation, and we have engineering principles and things that have come from science. And those things actually allow us to build satellites and launch them into space. But if we think about the fact that our planet is, in some sense, unique in that it requires a technological civilization with knowledge of the laws of gravitation and engineering principles to actually even do that process of launching satellites into space, it starts to make you think about it a little bit differently. And so David Grinspoon describes this process of a planet anti-accreting matter, in some sense, that actually requires intelligence or technology or knowledge to do that. So we don't see any other planets that we know about that are anti-accreting. Usually we talk about planetary accretion and planetary formation. Planetary anti-accretion requires an evolutionary history and acquisition of information to actually be able to do that process. So another example that I like to give about sort of how information might actually be physical is to think about the physical processes that actually make different elements on the periodic table. So people in this room are probably familiar with a lot of the kind of mechanisms that we get different elements, from Big Bang nucleosynthesis to the processes underlying dying stars. But we don't always think about putting technology as a physical process for some of these very heavy elements. But some of the heaviest elements, the only reliable process in the universe that can create them is a laboratory physicist. if you think about that as a physical process. So there are some things, like satellites being launched into space, or certain elements that require certain kinds of processes to happen before they can even be created in our universe. The laws of physics and chemistry alone can't do them. What we actually need is intelligent beings that understand those processes. And so David Deutsch has a nice way of talking about this. I like the way that David thinks about these problems. He says that base metals can be transmuted into gold by stars and by intelligent beings who understand the processes that power stars and by nothing else in the universe. And so when I think about what life is, I think there's two domains of physics. There's physics as we describe it, like processes that power stars, gravitation, and all of these things. And then there are physical systems like us that have some theories that we construct about the world that actually can do things with that and actually create new possibilities with that, like launching satellites into space or creating elements that are not excluded by the laws of physics as Schrödinger recognized, but nor can their existence always be explained reliably with that. You need an additional process. That's obviously all very abstract, so I'm going to get into more concrete science. So I do like to think about the philosophy of it. But what I wanted to get into with this is I've been talking a lot about technology and humans and the way that we interact with the world, because I think it's a manifestation of the physics of life. But that physics had to emerge with life. And so one of the ways that I approach the problem of life is trying to unify understanding everything from the origin of life to the subsequent evolution of the biosphere and what's happening now with our civilization as being part of the same physics. That's obviously a really tall order, but I think if we want to have a sufficient theory to be able to predict what life looks like on other planets, it's going to be at that level of abstraction. And so one of the proposals that I've been making throughout my career is that the origin of life transition is somehow related to this physics. And when we talk about the origin of life itself, we should be able to quantify the origin of life as somehow being a transition in information becoming a dominant mode of what the physics is happening in that system. So this being said, that information is with a caveat, because I don't think we actually really understand what this thing is. But there is some kind of transition where life emerges, and we have some kind of new physical processes dominating. And so in my field, people like to think about life in terms of defining life. So for example, a canonical definition that people give in astrobiology about what life is, and probably some of you have heard it, is life is a self-sustaining chemical system capable of Darwinian evolution. Who here has heard that definition? Who here, does anybody agree with that definition? I'm just curious. Okay. Maybe nobody's buying into it. Okay. Yeah, so I think one of the things that's difficult is that we are used to thinking, there's a couple problems with that definition. One is that it assumes life is chemical, right? And I think one of the things is that life emerges in chemistry. It's the level of physical reality where life starts to become, or whatever processes underlie life start to become prominent. But it doesn't mean that life is chemistry. And so if we take this sort of more abstract idea that life is something about information, it unifies the way we can think about the evolution of the biosphere from the origin of life to now, and that we can try to think about life not as an example of life, not as an individual cell, not as a chemical system, but as this process, this information processing structure that emerged on early Earth and has been expanding in space and time ever since. And you and I are one particular partition of that process. We're an informational structure that was generated by that evolutionary process, and we are part of that generating structure. But we can't really separate out individual parts and really think that the definition of life is a cell that reproduces. It needs to be part of that continuum. And so this is a very different way of thinking about what life is than people are used to. I think it's a really useful way of thinking about it when we're trying to tackle problems like the origins of life. Because if you want to talk about the origin of life, you need to understand what processes actually give rise to the emergence of life. And so the argument is there is some physics of information that exists in our universe in the same way that gravity exists in our universe. But if you want to study that, you need to study living systems. If you want to study the physics of life, the physics of information. Whereas in gravity, you might study something like a black hole. Right? So we are the examples of that kind of physics. And so one thing that comes out of this is that we might think of some systems as being more alive than others. So I am going to conclude the philosophy part because I think that's a lot of deep thinking about what life is. And what I want to talk about for the rest of my talk is actually what can we do with this set of ideas and how can we think concretely about how to approach even asking questions about whether this kind of physics exists, or how can we use any of these ideas to ask productive questions to try to get at this idea of what universal properties of life are. So I don't necessarily want to advocate that any of the ways I'm thinking about it are the right way of thinking about it, but I think one of the problems with the what is life question is people haven't known how to ask that question the right way because we obviously don't have an answer, so we need to open up new perspectives. And so the way that my group kind of thinks about the problem that I've talked about and how we can talk about it concretely is to kind of divide the work that we do into 4 areas. And I'm going to talk about a couple of them today, but not all of them. But I just want to touch on them briefly. So one of them is what is information in the physical world and how is it related to what we see as far as biological organization. So we do things like apply information theory to biological systems and try to see what unique patterns biology has that might distinguish it from non-biological systems. And how is the structure as far as the network structure or topology or causal structure of biology related to its informational structure. And the intelligence part is more about coarse graining and what coarse graining might be as a physical thing. And also is related to studies of consciousness, which I'm definitely not going to talk about today, but is really fun to think about. What I am going to talk about is a little bit of this and this idea of physics of living worlds in terms of thinking about chemistry actually as a representation of physics of life. Because I think chemistry is quite interesting because life obviously emerges in chemistry. And when we're thinking about what chemistry is as a physical system, it's an absolutely exponentially huge space. It's combinatorial large, and not every kind of chemical system can exist, but life does something really interesting in that space of what can exist. And then looking for life on other worlds, I'll touch on at the end, which is eventually hopefully going to give us some constraints on what we think life is. So I'm going to start with this question about whether life and non-life are actually quantitatively different and how we might think about that question. And so the approach is basically whether we can statistically distinguish a living system from a non-living system. And as physicists, I think we're really familiar with thinking about how to characterize physical systems from a statistical perspective. And I think one of the things in my field is that people haven't really adopted that, in part because living systems are complex systems. They have so many degrees of freedom, it's hard to think about what degrees to look at. But I do think that there's some merit to this approach. And so obviously we're familiar with statistical mechanics and the fact that we don't have to track every particle in a box to make some meaningful statements about what's happening in the box. We can talk about something like average velocity and talk about something like temperature, or we can talk about whether one box has a higher temperature than another box based on the average properties of the molecules and the distribution of their properties. And then when we actually adopt that kind of statistical framework, we're actually talking in ensembles. And so one of the problems that we have in biology is that we are no longer talking about simple particles, but the base level of biology is chemistry. And so we have to talk about molecules, which have a lot of structure, and they have patterns to the way they transform among one another, which is reactions. And so if we want to take a statistical approach to chemistry, we really have to start thinking in terms of network theory, which a lot of people in complex systems and people are thinking about physics of life think in terms of networks. And that might be the appropriate statistical framework for thinking about how we can characterize statistical patterns in life. And I think this is one of the reasons that network theory has been so successful in complex systems, is that it is an appropriate framework from just thinking about the fact that living systems are composed of many interacting molecules and that we can construct such a description. But then what we really need to do, what sort of the tall order is to think about what living systems are, and what are the measures on the ensemble of living systems if we could construct such an ensemble that would distinguish life from non-life. And so one of the questions that I ask on a regular basis is whether the ensemble of living systems is actually statistically distinguishable from that of non-living systems. And there's a lot of reasons that we need to think statistically in this regard that I can talk about at length. But I want to kind of dig into how we're thinking about it quantitatively. So one of the places where people think about whether you could develop a statistical mechanics for biology or not is in terms of something called regulatory networks.
I think you lost the microphone.
Oh, did I lose it when I... Oh, it's attached to my jacket. Excellent. Here we go. Let's see if my necklace makes a lot of noise now. All right. Good. So one of the places that you can start thinking about statistical approaches, or people have tried to develop a statistical mechanics, is actually studying gene regulatory networks. So we have genes inside our cells, they interact with one another, they form complex networks, and people have for decades studied the statistical patterns of those networks. The idea of representing those networks as Boolean networks was actually developed originally by Stu Kaufman, with the idea that there might be some kind of statistical mechanics of cell types, for example. So if we think about genes as being on or off, expressed or not, we can talk about their interaction patterns, and then we can actually look at the states, the attractor states of those complex networks. They should correspond to cell types, and they should have properties that we could statistically characterize, and talk about these bulk transitions in the attractor landscapes of these networks as being relevant to describing the complex interactions of many genes. And so what my group has done is actually really take that statistical approach one step further, where people had been applying, trying to understand these networks to what are called random Boolean networks. They basically were generating models from logic functions, Boolean logic functions, wiring them together and trying to model gene networks as random networks. What we did is actually take a collection of real biological networks that had been modeled as Boolean networks. So people for decades have been trying to make simple models of gene interaction networks and see if they could predict their properties or understand how robust they are. And so they construct these Boolean network models for a specific function of a specific small gene circuit in a cell. And so there's now a database called the Cell Collective that at the time of the work I'm going to talk about right now had 67 Boolean network models, but now has more than 70 that we're working with. And so one of the questions that we were interested in asking is, like, what kind of properties might be characteristic of the biologically functional network? So could we say, you know, this network is biological functional, is part of a living system, and this network is not? So where do biological networks live in the space of all possible Boolean networks? And so there's a lot of assumptions that go into whether Boolean networks are the proper representation of genes and things. But just building on this literature, we can ask real quantitative questions about it. And so one of the questions that you can ask is about the criticality of the networks, which is related to their information processing ability. So in complex systems literature, there's this idea of adaptation toward the edge of chaos, that systems can be chaotic or they can be ordered. So if you have a highly chaotic system, it's not very predictable in its dynamics. It can't store information long term because its dynamics are so unpredictable. And if you have a really ordered system, it is very rigid and non-adaptable. And so the idea is biological systems will be poised at criticality in between chaos and order because they can be adaptive but still store information relevant to the adaptations that work. And so it had long been conjectured that gene regulatory networks should be critical. Right? So a hallmark feature of biology should be that biological networks are critical in this way, that they actually exist at this edge of chaos. And so what we did to test that hypothesis was take these 67 biological networks that had been derived from real data as Boolean models, and we constructed random variants of them. So a Boolean network model has each gene modeled as a Boolean logic function, so that's a node in the network, and that determines how the interactions of the other genes, if they're expressed or not, update the expression level of that gene. So each one of the nodes in the real biological network has a Boolean function associated and some connectivity to other genes in the network. And so what we did was actually randomize it with networks that had the same causal structure, so they had the same exact topology, in terms of the number of edges, but the edges were randomized. So this network here actually has the same number of connections in the network, but it hasn't conserved the local connectivity of individual nodes, but it did conserve what's called the activity bias of individual nodes, so it conserves something about the Boolean functions in terms of how much they produce a 1 as an output. That's what the activity bias specifically is. And then we did another randomization where we conserved the local topology of each individual node, but randomized its connections. And then a third variant that actually kept the topology and had randomized it, but had an additional feature of using something called a canalizing Boolean function. A canalizing function has a particular causal structure in that if you specify one of the specific outputs, you always know the inputs, you always know the output of the Boolean function. So if you know one of the inputs, the output is always determined. And part of the reason that we did that is Stu Kaufman actually suggested that canalizing functions were preferred by biology because of their controllability and certain properties that they had with respect to cell attractor landscapes. And so we took an ensemble of each of these kinds of networks and compared it to the biological network. And so the idea here being this is the furthest away in terms of, if we think about distance in some kind of high-dimensional abstract network space of all possible networks, this is the largest ensemble, and each one of these is a sub-ensemble that's becoming more specific to biology. And what we did was use a measure of criticality developed by Ilya Shmulevich and Stuart Kaufman, which basically you perturb the state of an individual node, and then you measure how many nodes that it's connected to actually change their state under that perturbation. The idea being, if it's only one node that you affect for any given node, you flip its state, then your network is critical. So this idea is that if you have critical sensitivity, which we plot here as the average sensitivity of all nodes on the network, then your sensitivity should be 1, because that's the definition of a critical network. And what we find is that the biological networks are indeed critically sensitive, so they are critical networks. But these random ensembles are not necessarily critical, and most of the random ensembles are actually in the chaotic regime because they have a very high sensitivity, which means if I perturb the state of one node, it spreads very quickly and amplifies in the network. So it's very chaotic, the dynamics of these networks. But if we do control for the canalizing nodes and maintain the same causal structure as we see in biology, we reproduce the criticality of the biological ensemble. So the way of thinking about this is there's a very large space of all possible networks biology could exist in, and there's a space of critical networks. And the naive critical is that you can have critical networks that don't have canalizing nodes, but what we see to reproduce the criticality, specifically in the biological networks, is they have to have this additional property of having these canalizing nodes. And so criticality can be used to distinguish the ensemble of biological regulatory networks, and the reason for that is actually somehow deeply related to some of the ideas I was talking about at the beginning of the talk more philosophically and abstractly, that information processing is related to causation because it actually has to do with the specific topology and then the kinds of Boolean functions that are used within that topology and how the system is actually regulating the Boolean functions. And so we do see this kind of relationship between information, causal structure, the topology of the network, information being the Boolean logic functions, and the criticality of the networks in a very clear example here. And one of the things that my group is doing now is actually related this more formally to information theory. So we had done work before just on one specific Boolean network model of the fission yeast cell cycle, where we showed that information patterns as far as a measure of information theory, which is called transfer entropy between nodes in the network. Actually, the pattern of that over the whole network distinguishes biology from random networks. And what we're doing now is actually demonstrating that that property is related to the criticality of the networks because there's a deep relationship between sensitivity and transfer entropy, how it's distributed on these networks, which is forthcoming work. Okay. So I'm going to shift gears a little bit from the Boolean network models and thinking about gene networks and how we can think statistically at that scale to thinking about life across scales. And so this is going to be kind of shifting the specific examples of biological networks we're thinking about and how we're thinking about looking at this kind of physics of statistically distinguishing life from non-life. And somehow information, in whatever capacity we think about life being somehow related to that physics, is actually organizing matter, and how can we look at that in the patterns of networks that we see, to thinking about it more at the chemical scale. And so one of the points that I made earlier was that life is not a level-specific phenomena in the sense that I don't think the definition of life is chemical. It's not that life is a self-sustained chemical system capable of Darwinian evolution. I would make the argument that life is the physics of how information structures matter, and that process sometimes takes on an open-ended evolutionary process like we see in our biosphere. So I think the way Schrodinger framed the question is not quite right, because he asked, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? But I don't think the boundary of an organism is the right scale for thinking about the problem. And this is not to say that the way I'm going to pose it is the right way to think about it, but I think the way that we frame the question is really important, and we don't know how to frame the question. So this goes back to this idea that life is somehow a process that occurs across all of these different scales. But we know the foundational level of that process is chemistry, right? So inside each of our cells in our bodies are chemical reactions happening. When life emerged in the first place, it was in chemistry. And so one of the things I've been playing around a lot with lately is thinking about chemical space as the space of molecules and the transformations on those molecules and how we can think about what the structure of life is in that space and how life moves through that space. And just as one way of thinking about it, we know that when life emerged, certain chemistry was happening. By the time we got to fully functioning cells, very improbable chemistry was happening, right? In the sense that you are never going to statistically expect a protein to fluctuate out of a random chemical soup. A protein requires an evolutionary process to create it in the same sense that a table requires a history and information to create it. So there's some boundary in chemical space that we get to that life crosses where you get into very highly improbable or unlikely chemical structures to emerge, and they require some kind of information for them to actually stay in the system, to reproduce or to be copied, and to propagate. And that expansion of chemical space is really interesting in terms of the fact that biology has been inventing new chemistry as it's been creating higher levels of organization through its evolution, to the point that now with modern technology we can explore more of chemical space than the universe probably has been able to do without intelligent beings like us. And maybe this happens on other planets, but we're inventing pharmaceutical drugs and AI to explore chemistry at an exponential rate. So if you think about the process of what's happening in terms of what molecules can exist and how they exist and what kind of transformations are happening to make them exist, all of that is happening at the level of chemistry, but there's some signatures of this entire evolutionary process on top of it. So we're interested in that. And so to go back to the idea of thinking about a statistical framework for thinking about chemistry, now we're talking about how do we compare the ensemble of living chemistry to non-living chemistry, and is this even a meaningful statement? How can we get at this? And so one of the questions that we've been asking in my group is whether an ensemble of biochemical networks is statistically distinguishable from non-living chemical networks. And so one motivation for this is actually in terms of astrobiology and thinking about biosignature observables is that we know life is an emergent property, and so therefore its quantitative observables should also be somehow the emergent property of complex systems of molecules that are interacting. And so what I want to know is what are the appropriate coarse grainings of this network, basically what are the statistical regularities that we should expect in biological networks. So how we get at that is to actually sample the ensemble of all possible biochemical networks. Now that's obviously easy to say, very hard to do. And whether we could even say we were sampling a tiny infinitesimal fraction of it is quite difficult. But we do the best we can based on data available. And so what my group has been doing is basically scraping biochemical data or bioinformatics databases to get genomic data. And for the work that I'm going to talk about, we had at the time 21,000 bacterial genomes, 845 archaea, and 77 eukaryotes. And then we also wanted to look at ecosystems and the biosphere as a whole. And I'll explain a little bit about the motivation for this. But the ecosystems, we had a large database of metagenomes. So metagenomes are just when you sample a genome from a community and you don't really have knowledge of the individual. So if people go out in the environment and they sample genomic information, it's usually metagenomes that they're sampling. Like when Craig Venter was sending things all over the ocean. And then the biosphere we consider to be 8,600 plus known reactions that biochemistry does that are cataloged in a particular database called KEG, the Kyoto Encyclopedia of Genes and Genomes. And so what we do from this data is actually take annotated data of the specific genome, cross-reference it with another database that has the chemical reactions that those genes are actually encoding, and we construct networks of what reactions that organism or ecosystem can catalyze. And so the idea is that we can build an ensemble of biochemical networks across scales, considering both individuals and ecosystems, and the planetary scale organization of life, and look for statistical regularities. And so one of the things that I like to play around with is to think about the what is life question, not at the level of a cell, but actually at the level of a planet, and how that planet has partitioned into individual life forms that we call individuals. Because species change over time, individuals change over time, but there is a process that we call life that has persisted on this planet for 3.5 billion years plus. And so we can kind of abstract thinking about the physics of life at the planetary scale to thinking about planetary scale networks. And if we want to ask this question about the ensemble biochemical networks and is it statistically distinguishable from non-living networks, what we're trying to ask is whether there are actually regularities across these scales. And so the way you can ask questions about that, obviously, is to look at scaling laws. And so the way that we've been framing it is to think about the size of the chemical system in terms of the number of compounds or molecules as being the parameter that we're looking at scaling behavior relative to. And on the y-axis, we might look at some network property. It doesn't have to be a property of the topology of the networks, the connectivity of different reactions and nodes. It could also just be statistics on the properties of molecules. It could also be properties of just the number of reactions. So there's lots of different things that you could look at. And part of it is we don't know what the parameters are or what things are actually might be distinctive of life if there are any things. And so with the initial work that we were doing, we were testing 3 alternative hypotheses. One is that life is a level, or the organization of chemical networks is level specific. And so the idea being that if you looked at individual organisms and their biochemical networks and you looked at some of the properties of the scaling behavior, it would be different from ecosystems and the biosphere wouldn't be anywhere on that curve. And if we looked at random chemical systems, if we could construct abiotic systems or random chemistry, it would have a very different behavior. We might also alternatively consider that the scaling behavior that we observe is a universal property of chemistry, in which case we would expect all the networks to behave the same way, independent of scale, independent of whether they were living or not. And this would be sort of the null hypothesis against my conjecture that living systems have different statistical properties. Or we could say it's a universal property of biology, but to validate that, we would at least need to demonstrate that individuals, ecosystems in the biosphere have regular scaling behavior across the 3 levels of organization that we're looking at. Obviously, we only have one biosphere. If you ideally wanted to do this, you would take chemistry from many planets. And then if you compare it to random chemistry, that has different scaling behavior. So we actually did this for our 28,000 or so networks. So we constructed networks. We did the statistics over the topology of the network. So basically counting things like how many reactions does a molecule have, which would be like the mean degree of the molecule, or things like average shortest path length, which is shown here, which is the shortest number of steps in the network between any 2 molecules. Clustering coefficient is like how many molecules share neighbors in the network. But we looked at a bunch of different properties as a function of the number of compounds in the network. And shown here is in blue all of the individual organisms. So this is the 22,000 or so bacteria, archaea, and eukaryotes. And you can actually, so we have a lot of redundancy in terms of biochemical diversity because there's a lot of sampling of organisms from the same phyla, for example. And so if you remove that redundancy, you still see very regular scaling behavior. So that it's pretty robust to a lot of different kinds of tests on these data sets because biochemical data sets are notorious for having incomplete data or oversampling organisms you can culture and a variety of things. But if we look at the individual organisms and then the 5,500 ecosystems, we see very similar scaling behavior. And when we also look at things that are independent of the actual network structure, like number of reactions or number of enzymes encoded in that gene or genome, we also see very regular scaling behavior because some of these things can be very sensitive to the network projection you choose. We did multiple network projections for that reason also. And something interesting is some of these scaling behaviors are statistically distinguishable between ecosystems and individuals, and some of them are not, which says something about the architecture of biochemistry across scales. So we do observe at least some kind of universality in biochemical network structure for the data set that we're looking at. But to test whether this is actually like a property that we should start to pursue to try to understand statistical regularities in life, we have to at least demonstrate that it's not a property of random chemistry. And that part's actually a lot harder. So with the Boolean network models, I was talking about randomizing the networks to construct random variants to test the hypothesis about what was unique to biology. We effectively ended up having to do the same thing here because it's very difficult to build a proper control for a biological system to say the system's not alive, the system's alive, how can I make an effective comparison between them? So what we end up doing in practice most of the time with the work that we do in my group is to actually look at randomizing properties of the networks that we do have. And so one thing that we know about biochemistry on Earth is that all organisms share a core set of reactions that are associated with core functionality of life. We have a last universal common ancestor, which all life is believed to have descended from, and those reactions are present everywhere. And so what we tried to do with our sampling to construct random networks, so shown in gray here is the actual biological data. And what we did with this orange curve here is take the reactions that are in the Kyoto Encyclopedia of Genes and Genomes, the ones that I identified as being biochemical because they are known to be catalyzed by an enzyme, and constructing networks randomly sampling those reactions. And so that's what's shown in orange. So if you randomly sample reactions to construct networks and you look at their topological properties as a function of size, they exhibit statistically distinguishable scaling behavior. You can tell the difference between one of these artificial organisms, biochemistry, and a real organism's biochemistry, which is perhaps not surprising, but is useful because we're starting to get at quantitative ways of talking about that. And we then did a sampling to try to see if this was biased, if the reason that we were seeing the particular scaling is because of the reactions that are common in life. So what we did in this case is sample reactions by their frequency that they actually occur across living organisms. So some reactions are really rare, they happen in one organism, and some reactions are very frequent, they happen in every organism. So we generated a frequency distribution based on the reactions and constructed random networks. So these networks statistically should follow the same distribution of reactions over organisms as real biology, but they're not functional biological systems unless we actually went and built them in the lab and tested that. And those ones exhibit much closer scaling behavior, although in some cases you actually still can distinguish them. And just to generate another ensemble for comparison to biology, for the ecosystems, we constructed what are random ecosystems where we took genomes, like whole genome networks, so individual level networks, and merged them to make ecosystems. And those have similar scaling also to the real biological networks, but for some measures you can still statistically tell the difference. So we can verify, at least with these kind of computational experiments, that biochemistry has properties that statistically distinguish it from non-living chemistry. And this is just to show that you can do the same thing for a number of enzymes, so a number of reactions in the network. And so one of the main take-homes is that you can do the statistical comparison, but I also like to make this other point, which I think is less obvious from the actual results of the work, but kind of an interesting implication as far as hypothesis building for future steps and actually understanding origin of life process, is that actually explaining the scaling we observe requires individual units that share reactions, right? Which seems kind of like a really obvious statement to make, but it's quite interesting to me because we have this thing that we call the last universal common ancestor, which is this idea that life descended by an evolutionary process from a common ancestor. And the traditional narrative there is that ancestor outcompeted everything else, and then life evolved from that ancestor. So it's kind of like an evolutionary bottleneck. But you might have an alternative process that there's actually constraints at the ecosystem level that organisms need to share a certain amount of reactions. And if you had some kind of collective evolution process that was some kind of like statistical evolution, people have made collective evolution models for early life for genomic evolution, that the reactions themselves actually would have had to lead to constraints on that global topology at the ecosystem level, which enforce individuals to have this property of universality in terms of some of their chemistry. So this is actually a hypothesis about the origin of life process itself that I'm actively interested in testing and building into a framework for thinking about life as a planetary scale process in terms of the chemical composition and the structure of chemistry and how that process has evolved over time. So just to kind of continue on this, I just want to talk, so that work was published about the scaling of the reaction networks, but I want to talk a little bit about other ways of thinking about bringing in new approaches to thinking about statistical regularities in biochemistry. And so one of the things that we're doing now is actually to take this biochemical network and label all molecules by whether they're chiral or not, and also how many chiral centers they have. Which is, chirality is obviously a really interesting property of molecules, that molecules can sometimes have mirror images that are, just the exact same atoms, but in the mirror conformation, and then you can't actually superimpose them. And chirality is actually deeply important in biological systems because all of the proteins, for example, that are in your bodies are composed of only left-handed amino acids. So we think in the origin of life there was some symmetry breaking event where we had left-handed and right-handed amino acids, and then biology, through some evolutionary selection process as it emerged, selected only left-handed amino acids. What we're doing here is actually trying to look at the statistics of how chirality is distributed in biochemistry writ large and how that actually leads to the structure of biochemical networks in chemical space. Because chirality is interesting for a couple of reasons. One is that you're more likely to get chiral molecules with higher mass molecules. So the higher the molecular weight, the more chiral centers you have. So the number of molecules of specific mirror images of a molecule actually increases with molecular weight. So there's some kind of scaling associated with that. But also it places constraints in reaction space as far as like entire pathways of reactions. Once you break that symmetry, you break off a possibility. So it's about symmetry breaking at the network level and how biology actually mediates that. So we're interested in it for a lot of reasons. This is just the biosphere as far as where achiral molecules labeled in red and chiral molecules are. One of the things that we've been able to demonstrate so far with this work, just as sort of like a brief interlude, is that 75% of molecules in biochemistry are chiral across the board. So it seems to be like a regular feature of biochemical networks, even when we randomly sample reactions. And so we do think that there are some principles to uncover as far as chirality and how it's leading to the emergent network structure we see. So that's a property of molecules, and that's starting to bring in cheminformatics approaches to thinking about biochemical networks and their properties. But we can also think about the enzymes themselves and their functionality. And so when people do these reaction databases, they have enzyme classes, major enzyme classes in the database. It's called enzyme commission number that you can actually look at. And there's 6 major classes of enzymes, which are associated with major functional groups of enzymes. So oxidoreductase catalyze redox reactions, ligases catalyze binding, hydrolases actually hydrolyze molecules. So these are different functions that enzymes can do. And if we look at the scaling behavior of each of the enzyme classes as a function of the number of compounds in that organism or ecosystem, we see very regular scaling behavior there as well. So it's like biology is basically using regular statistical ratios of the different enzyme functional classes across levels of organization. There are distinctions between, obviously, like eukaryotes and their scaling behavior, shown in yellow here, and bacteria and archaea. But across the board, we see very regular behavior. And interestingly, ecosystems have some differences from individuals with respect to oxidoreductases, which are associated with energy acquisition. So there's some different scaling that emerges at the level of ecosystems related to how energy is harnessed and used. And ligases are really associated with like universal function associated with DNA and RNA structure and how we actually build like translation machinery and things. And so that universal, those reactions are believed to be universal across organisms, and so we see them actually sloping off in their diversity and scaling as a function of network size. So the main point is that both in terms of the topology of the networks and the statistical properties, but when we dig into properties of molecules at a certain coarse-grained level and also properties of reactions, we can actually statistically predict properties of biochemical systems. Okay. So the last part I want to talk about just for the last couple of minutes is how can we use any of this to discover examples of alien life. And so I mentioned in my deep philosophical dive at the beginning about this idea of trying to understand how much of the universe was actually complex or living matter. And I think one of the things that original life field needs and astrobiology needs, and also like thinking deeply about physics of life, is a tighter integration of theory and experiment. And the way that it has been very successful in physics when we build large experimental machines like the LHC, but we always have really strong theoretical motivation and we have theories driving things. We need that feedback when we're thinking about the origins of life. And I do see some like hope for these kind of approaches starting to develop. So there's been some really great things happening in the original life community in the last decade, tying more with artificial life approaches and thinking more about this idea of what's called messy chemistries, which is taking more like random chemical soups and trying to just see what their properties are under different environmental forcings. So I work with one lab that's doing that kind of approach. which is Lee Cronin's lab at University of Glasgow. And one of the things that they do is they take what's called like soup chemistries of different molecules that might be present in an environment, say on prebiotic Earth, and they subject them to different environments. So you might add a mineral or change the pH or change whether it's hydrated or dehydrated. And what they can actually show is that you can actually distinguish the chemistries based on their history. So if you have different orders of mineral addition, you'll never get the exact same product distribution because of the stochasticity of chemistry and random fluctuations. So you can't, if you take a chemical mixture and you try to let the reactions happen and then you add some mineral catalysts and things, it's not exactly predictable the exact distribution of products you're going to get because there's chemical space is huge, there's random fluctuations, and some of the molecules might catalyze reactions and others not. And so you get a lot of variation. But what they've been able to show is that you can actually at least distinguish where you are in product space at a coarse grain level, at a statistical level, based on the different histories. And so one of my hopes is that they're actually doing this in sort of an ensemble approach because they have robotics equipment in their lab for evolving these soups and they can do many different histories in parallel, is that we might start doing things that are more like what the particle physics community does, where they think about bounding the probability of processes that happen in the universe. So this is a picture of Super Kamiokande, which obviously one of its goals is to try to bound the lifetime of a proton. And so when we don't observe that event happening, we can at least bound its likelihood. We have no statistical approaches for thinking about origins of life because the way origins of life experiments are conducted right now is somebody has their pet chemistry that they like to study in their own lab. And we're not thinking in terms of statistical properties of chemistry or building large ensembles. And so I think what we need to do is actually build this kind of scale experiment and look for interesting lifelike chemistries emerging or not emerging and at least bound the likelihood of that process. And so one of the things I also think about quite a bit is how to actually start thinking statistically about looking for life on other planets. And so there's a big problem right now in the astrobiology community about thinking about looking for life because we know we have a huge diversity of planets, of exoplanets, and we're looking for the Earth 2.0, which we don't know if Earth 2.0 exists because Earth exists the way it is because of life. So looking for an Earth-like planet is a little bit difficult because we don't know how common life is. But we are looking for planets that have Earth-like properties like similar mass and water, but we don't know what kind of worlds life is likely to exist on. And so we're losing a large fraction of the potential planetary space that we could be looking at. So there's been a lot of talk in the exoplanet biosignatures community, the people are looking for life on exoplanets, about how we should search for life. And a lot of that is about what kind of signatures that are remotely detectable can life produce. And so the qualification there is it both needs to be something that's a signature of life and remotely detectable, which is quite difficult. And what the community that works on exoplanet biosignatures has been realizing in the last decade is there's a lot of possibility of what are called false positives. So you might ask, well, if we're looking at the spectrum from thousands of light years away or thousands of light years away, how are we possibly going to be able to distinguish whether that planet actually had that spectrum produced by life with those specific molecules, or if there was some abiotic process on that planet that actually produced similar spectral features. And we know now that dead planets can fool us because there's a lot of mechanisms for producing canonical biosignature gases. So one of those products that we think about is oxygen. Oxygen in our atmosphere is a product of photosynthesis. And so we know that oxygen is very abundant in our atmosphere because of biology. And for a long time, oxygen was thought to be a biosignature on exoplanets. But then we realized there were all these other kinds of mechanisms on different planets that could produce oxygen-rich atmospheres. The most obvious one is just if you have a water-rich planet in a high UV environment, water can photolyze, and then you basically get oxygen, and the hydrogen escapes, so you get an oxygen-rich atmosphere. And so the community's been trying to think about ways of trying to get around this false positive problem. And so one of the ideas that's been developed is try to think about detecting life statistically. So we have limited data, and we need to be able to actually infer the existence of life on planets. And so I've been developing with a lot of my colleagues in astrobiology that work on exoplanets new statistical frameworks for life detection. And one of those is actually based on the same idea of quantifying statistical properties of networks related to whether you could tell the atmospheric of a living world from a non-living world. And so the idea there is that if you have a planet with life, the statistical patterns and the interactions of molecules in its atmosphere are going to be different than non-living planets, and we should be able to infer those differences remotely. And there's a long chain of things that you have to do to develop this kind of approach and actually do it rigorously. So the way that we're starting it is actually to think about Jovian atmospheres, which have nothing to do with life, but they have everything to do with thinking about remotely detectable observables right now. And so we've been developing network theory as a tool for understanding disequilibria in planetary atmospheres. And what we're finding is if you actually look at the properties of the chemical networks in the atmospheres of remotely detectable planets like a hot Jupiter orbiting another star, you can actually, even within the limits of observational uncertainty, tell the differences in drivers of disequilibria based on the network properties of the atmosphere. And the idea being that when we move to terrestrial worlds, biology will be one of those drivers, and we might use this as a biosignature that is statistically robust, doesn't depend on assumptions about the kind of molecules biology is using, like oxygenic photosynthesis does, and we can use it as another mechanism of adding to confidence levels for biosignature detection. Which gets to this point that we're thinking about the biosphere as a network and the atmosphere as a network, but you might think about the physics of living planets and chemical space as these coupled networks evolving through time. And I don't really know about the noosphere, but I know technology is influencing the dynamics of this in many ways. So that's kind of future directions for that. And I'm just going to conclude with my last slide that this is how I feel every day when I think about physics of life. And it's really exciting because there's a lot of questions and not very many answers. And I do want to thank my research group because I have really brilliant students and postdocs that I get to work with every day, and they're amazing, and also our funders. So thank you.
All right, do you have any questions?
How do you know when you've answered the question?
I think we will know when we design an original life experiment that we can actually see is successful. So my personal view is that we're not going to detect aliens on another planet. What we're going to do is evolve alien life in the lab from scratch in some kind of major experiment that's an international effort of the scale that I described.
What makes that life alien as opposed to, say,
It's actually, technically, it's not alien because it's still a product of our biological lineage because we generated the experiment to create it, but it's the closest you could get to alien life. So I think the thing is, like with origins of life, what is really hard is removing our agency from the process, and this is one of the things that's very difficult about the way original life experiments are constructed right now, is they have a lot of teleology or agency built into the experiment in the sense that I'm in the lab and I set up the steps and the conditions to make this one molecule. And so, so I think what we need to do with original life experiments is just remove that as much as possible and see if we can at least kind of bound the probability of getting things that we expect to be lifelike within that space. But I think, like, when I think about the future of astrobiology, I think much more like how physics has progressed, and I'm envisioning like large-scale, massive chemical experiments that are doing this kind of thing coupled to exoplanet observations, where you're actually using planetary parameters from exoplanets in these massive experiments. But to actually do that is like a 50 or 100-year effort. So I think the problem is really hard, and I think the problem I see with my community is we're not really appropriately tackling how hard that is. We're convincing ourselves that synthesizing an amino acid under prebiotic conditions is doing origin of life science. And it's relevant, but it's not the origin of life, right? And I think that we really need to really think critically, and I think it's a really important frontier in fundamental understanding. It needs to be done. Yeah?
The word cell popped into your talk a few times.
Yeah.
Are there current examples of life that does not have a cell membrane around it?
I don't know if, so there's, well, one example is like, yeah, no, there aren't, but there are things that might be like rough approximation, like they don't really exist, but people talk about like use cell-free extracts and things, so they actually have like biological functional components in a test tube, but you still have a boundary. So all life, as far as we know, is cellular, but I think part of my point is, so I think boundaries are important. I didn't mean to imply that. I think individuals are critically important, but I think the reason individuals exist as they're partitions of a larger system. So a cell is like the current physical representation of a biological lineage when you look at it at one instant in space and time, but that is actually part of a much longer extended structure if you want to explain that cell. And I think the difference is, do I want to talk about the cell as I see it and exists now, or do I want to explain the cell as like from a and I think you just have to have a different reference frame for that. But boundaries are important.
Can I ask if
Yeah.
you were given $1 billion to design the origin of life experiment, would it be one giant experiment over decades, or would you scatter the money around many different approaches?
I think I would scatter it, but I would do it in a way that it was building towards something that was unified. And I think
What would the big one look like?
I think that would have to come out of the process of developing it, but I think a lot of it would be developing more robotics and approaches for doing chemical experiments, more ways of doing analytics for analyzing large, messy chemical data sets, and more theory that actually tries to embed ideas from like abstract theoretical physics into how we think about mapping out that chemical space and what information is doing in that space. And planetary, like, I mean, the scope of it, I could map out, I could write it, yeah. but I think,
$1 billion, so.
yeah, I think, yes, please give me $1 billion. No, I think the thing is, the community internationally has a lot of really good ideas. It's just not focused in the same way as, say, like the particle physics community is internationally on, like, what's the big question and how do we get at it? And it's just because our field hasn't thought that way. And I feel like if I have one soapbox, it's just to convince them to think that way. It doesn't have to be the way I think about it, but we need to do something.
One last really good question, yeah.
Do you envision any other type of life based on any atom but carbon?
I think that's a really interesting and difficult question. I mean, people always make the argument silicon, but like low temperature. And I personally, I think the way I think about the problem, carbon is the most likely candidate. I think once life emerges, life might copy itself to other substrates in the same, like in the way that we're building all kinds of things, but I don't know if life can emerge without that kind of diversity of molecular structures, which is a canonical argument people make, but that's sort of, I think there's legitimate physics behind that as far as thinking about combinatorial space and possibilities and things. But I don't know.
All right, let's thank Sara again.
Thank you.